Afternoon, everyone. I'm sorry that I couldn't go to Corsica, but I'll present our work on microbiomes and the development of next generation sequencing methods for analyzing microbiomes in the hosts that have been dealt with in Tropicsafe, that is palm, grapevine, and citrus. Today, of course, I'll focus on our work on citrus. So the purpose of our work was to develop a next generation sequencing based method for one, exploring phytoplasma or Huanglongbing, genetic diversity in single hosts, and that could be, or that would be plant hosts or insects. And the other purpose of our work was to explore Libéribacter interactions with other members of the microbiome, that is, other bacteria living on or in the plant. So first, a little bit on next generation sequencing, or metabarcoding as we also call it. So in our lab, we are dealing with a lot of different types of samples, from soils to plant roots, or even air samples, as you see in the middle, and plant seeds, but of course also plant material, including material that could be infected by phytoplasma or Huanglongbing. I just need to see if I can start my pointer.
Yeah, the pointer is there.
You can see the pointer.
Yes, we have the pointer, we see it.
Super. Okay, so I'll continue.
Thank you,
So we deal with a lot of different samples, and from those samples we do a DNA extraction, just a standard DNA extraction of genomic DNA. And from there we do a PCR amplification, and we have primers specific for nematodes, fungi, oomycetes, and bacteria that are able to amplify target sequences within the genome of the target organisms. So typically we amplify 300 or 400 base pairs or something like that. Also included in the primers, we have what we call tag sequences, which means that we can distinguish the PCR products after the PCR, so we can actually pool up to 500 samples in one sequencing round, which means that the technology actually becomes quite cheap. So after PCR amplification, we do a sequencing on a Illumina, what we call amplicon sequencing, on a MySeq sequencer. And by sequencing in this way, we are actually able to obtain, say, 10 million individual sequences from the amplification. That is, we are able to identify up to 10 million species in the samples. Of course, less if we pool samples. And then we have different ways of analysis of the data, and I'm just here showing one way of doing a network analysis, but that could be anything of identification of the individual species and the relative abundance, and etc. and etc. So, however, there are some. We realized after having looked into phytoplasmas and Libéribacter too, that there were some challenges with those organisms. The first one is quite universal, that when you want to amplify bacterial DNA in a plant background, you usually, or you have the risk of amplifying a plant organelle DNA like chloroplast DNA or mitochondria. So that's one thing you have to consider when developing a NGS method for plant living organisms. And the other problem was that the standard NGS primers that are usually used for bacteria, they do not actually, I know, they do not amplify phytoplasma DNA, and also there might be some problems with the Liberiobacter DNA. So the first thing we did was to look into standard primers that are already on the market or published or whatever. And we found one primer pair, this 799-1193 primer pair that amplifies the V5, V7 region of 16S rDNA. However, if you look to the right, maybe it's a bit difficult to see, but that's an alignment of a lot of 16S sequences from phytoplasma and Liberiobacter, and in the for the reverse primer, and you see that there are some nucleotide differences in this region. So we were sort of forced to make a new primer, and that's what you see here in the bottom with the red nucleotides, which are different from the original primer and which are having some degeneracies. We were quite, or rather, that was the best we could do here, and then we tried this on different plant samples. And I just show you one example here of an amplification of samples from different hosts, and that this is a mixture of citrus and palm and grapevine, as far as I remember. And you see, it's not a beautiful PCR because we use quite low amplification temperatures to sort of be sure that we catch everything. So what we do after the amplification is that we pool all the samples that all have a different tag, so we can distinguish them later by sequencing. So we pulled those and ran on a gel, and then we cut out the band of interest, which is here a little bit more than 500 base pairs. And then we did the sequencing and the data analysis. So what we have looked at so far in the Tropicsafe project is shown here. We, of course, also got samples of grapevine and palm. And I would say that numbers are not, I mean, it's not huge numbers. So you see, we had, like, I didn't calculate, but around 50 samples in total, which is, of course, nice and sufficient to develop methods. But for a very deep data analysis, it's on the lower end. So what did we see? And now I'm also showing the data from grapevine and palm and some insects we got, but focus just on the first column, which is the one on citrus. And you see, this is a relative abundance of all the sequences from all the samples of citrus. So it's sort of an overview. And I've put an arrow here with the Liberiobacter reads, which are up here, number 2. But in addition, we saw a lot of other bacteria in the plants, like Acinetobacter here, and then we saw this is a sort of a bulk of all the rare sequences, so that's not really so interesting. And then we also saw some other species. But compared to, if you look at the relative abundance of Liberiobacter in the samples, it's much lower than, for instance, the phytoplasmas in grapevine. Yeah, I think that's all from this slide. So, looking a little bit closer on the composition of the microbiomes in the different locations. As you see, we got samples from Cuba, from Guadeloupe, Jamaica, Mexico, and South Africa. So that was a huge difference. And here an arrow is placed with the Liberiobacter sequences or reads, and also here for Guadeloupe and Jamaica here. So we did find a number of reeds from those locations, but actually from Mexico and also South Africa, we didn't find a lot of. South Africa is this small fragment here. We didn't find anything in the Mexico citrus, or at least not in this enough to be presented in this bar chart. There might be a very low abundance. So looking at the insects that we obtained from collaborators in Tropicsafe, we could see, or actually we couldn't see any Libera sequences in the insects from Cuba here and also from Mexico. However, we did in the this batch of insects of Passifloraestis or something like that. I don't know. We didn't find much Liberibacter, but actually we found some phytoplasma reeds. So this is the data presented in a little bit different way on the Candidatus Liberibacter samples. So you see, again, it's separated into Cuba, Guadeloupe, Jamaica, etc. But to my view, the distribution of reed abundances is similar across the different locations. So what we see here is that some samples, you see up here, have 10 to the, I mean, that's around 1,000 reads within one sample of Liberibacter sequences. So we have the whole distribution from very, very few to up to 1,000. I would doubt a little bit the very low sequence abundances, like I think this is maybe 1 or 2 sequences, which might be a contamination, or at least that should be confirmed. We did include replicates, so that can be done. But this shows a little bit of the distribution of the abundance of Liberibacter within the different locations and in the different samples. So we also looked at what we call the beta diversity of the citrus samples. That is the similarity of the bacterial communities within the samples, meaning that if we have one citrus sample, we look at that might be represented in one of the dots over here. That might be one citrus sample. So we did this analysis on the different locations again, and you see there is some clustering, maybe not so surprising. I mean, here we have the Mexico citrus, and we also see a clustering of the South Africa citrus. And up here, for instance, you see the Guadeloupe, which is a little bit more spread, but we also got more samples from there. So this tells us that the bacterial communities are more similar in one location compared to another location. Again, maybe not so surprising. This might be, of course, caused by location differences, you could say, but we don't know if there are different cultivars of citrus being grown in the different places, and etc., etc. But at least it shows that there's some logics in the way samples are distributed. Yeah, okay, then a little bit more complicated way of analyzing the samples. The dots here represent individual species found in the different samples. So one dot could represent, we call it OTU, operational taxonomic unit. So that could represent one species. And of course, you cannot, from these very complicated figures, you cannot really figure out what is going on. But what we see here is associations between the different species. So, and down here we have the pink dots. Here are different Liberibacter sequence types, and there seems to be some associations between, for instance, Metulocella and Metulobacterium. green ones and the orange ones. But of course, we can see this in tables and so on, how the correlations are. And if you look at the right side on the other figure, we have visualized the positive correlations between individual bacteria. This is sort of all the bacteria that we found in the samples, and the pink ones are the Libibacter. So again, the gray lines, they're positive correlations, and the pink lines are the negative correlations. So just to show you that there is a potential, at least here, to being able to identify potential antagonists. Okay, a negative correlation, of course, doesn't imply that there's an antagonism going on, but at least that would be the organisms I would look at in other bioassays or by other methods or by PCR, whatever. There might be some correlations going on here. So, in conclusion, I believe we have developed a next generation sequencing based method that could be useful for investigating Libibacter interactions, or could, phytoplasma if it's in that host and other hosts, with the total bacterial communities. And I also think that this study of the networks, the last slide I showed with the correlations so on, could help in understanding the ecology of plant pathogens, or Libibacter in this case. And also finally, the negatively correlating interactions we don't know if it's an actual interaction, but that could imply antagonistic effects and could lead to improvement of Libibacter disease control in the far future, I think. So thank you very much for your attention.